Swayam Prabha. Digital India. Educated India.
Now, we propose one more parameter. This will constitute the residues which have more number of contacts. For example, if there are many clusters, right, one residue is forming many long range contacts. So, then see in a protein how many clusters you can find, right, based on long range contacts, and there are more clusters are formed with long range contacts, then these proteins take time to fold. Under this assumption, we see the residues which can have more number of contacts. For example, take this as an example. How many contacts? 4 contacts, right, here only 4. So, if you take this one, 8 contacts, if this one 11 contacts. This is for the residue T1. For example, a protein, one case only 5 residues have these multiple contacts. One protein they have 20 residues which have multiple contacts, which will fold fast, which will fold slow, right. One with only 5 residues they are having multiple contacts, another one 20 residues have the multiple contacts, which one will fold fast? The 5 one will fold fast, because only with the 5 one they have to get more number of contacts. In this case, all the 25 different residues they are making the contacts from different residues, so it takes time to get all the contacts. So, here is one example. So, we have the different classifications alpha beta mixed and the fast and slow folding proteins. So, here this is the percentage of residues which have multiple contacts. So, if you see the fast and slow folding proteins with the at least with the condition of 5 residues, if we see in here we see the clusters only if the residue contains more than 5 long range contacts. If you do so, the fast folding proteins there are only 12.6. and the slow folding proteins you can see the 22.6 percentage of the residues they are present in the case of slow folding proteins. This will tell you that the proteins with more number of the multiple contact residues are slowing down the folding process. Then I show another example. We take the fastest, that means ln kf is more than 9, and the slowest is less than 0, right. We have 8 examples for the fastest and 7 for the slowest. So, here if you see only 6% in the fastest and more than 25% in the case of the slowest proteins. This will clearly tell you the proteins, right, which containing more number of residues with multiple contacts, right, they are slow folders. Here we did with the 47 fast and 28 slow proteins. Here the fastest and slowest, we can see the difference between the percentage of residues with the multiple contacts. So, based on this information we can derive a parameter, just we can relate the folding rate within because we need to quantify, right. Here this will tell you some numbers, how many residues which you have with threshold of more than 5 contacts. In the here we give the more than 10, we can change this number. Then also in this case we need to quantify. So, based on that there is another parameter called the multiple contact index have been developed, right. Here they take distance between residues, this is already used in the previous methods, right. For example, contact order, long range order, and the second one is sequence separation, this was done earlier. Now, here in addition to that we added one more feature. What is this feature?
Number of residues.
Residues with multiple contacts. Okay, for example, if you see this figure, here the how many this you take the T1, how many long range contacts this can form? 1, 2, 3, 4, 5, right. So, in this case n Ci equal to 5. If this is the case, then you can define n mi equal to 1, because if it is more than 4 or 5 you can put any cutoff, right. And if this is the case, MCI you can calculate sigma n mi by n for all the protein and get the number. Now, see whether this has any influence in the folding rates compared with long range order and the contact order, right. Here we have different data sets, right, 2 set proteins and 3 set condition, 3 set proteins, right. So, these are data. You can see this is a 2 set proteins, right, 50 proteins, contact order is 0.6, long range order is 73, this is TCD is a total contact distance, that is the multiplication of CO and LRO, right. So, here is also 0.73, and MCI is 0.8. Okay, this information will enhance the correlation between the MCI and the folding rates. So, now we try to see whether this is correct or not, how far this can be trusted. So, we set another 27 two-state proteins, same experimental conditions. The beginning of the lecture I discussed about the data determined by the different experimental groups, the same conditions. So, for that if you do that, there also you get about the similar correlation minus 0.81. That means, MCI is a good measure to relate protein folding rates. Then we use the same with the 3-state proteins, here also we could get the same correlation of 0.83. So, then this can be a good measure for predicting or relating the folding rates. Then we need to compare the 2-state and 3-state proteins. There is one important information between 2-state and 3-state proteins. The 2-state proteins we need to normalize with n, for example, the long range order. So, what is equation for long range order? Nij by n. But the case of 3-state protein, right, if you normalize the values is very less, but if you do not normalize then this is 0.83. Still it is very clear why this normalization is very important for the 2-state proteins and it is not required for the case of the 3-state proteins. 3-state proteins if you normalize the values are very less, 2-state proteins if you do not normalize the values are very less. So, it is very important to normalize the 2-state proteins, right, to get the folding rates with better performance. So, till now we discussed about the different features, contact order, long range order as well as the multiple contact index. Now, if we see whether any difference in the folding rate and the number of the fast folding proteins and the slow folding proteins based on the number of contacts. Here if you see the x axis number of contacts, y axis the percentage of residues, right. So, the percentage up to the 7 residues, for example, up to here. So, see this higher in the fast folding proteins, after that it is higher in the case of slow folding proteins. The same in the case of 27 2-state proteins and the 3-state proteins as well as another set of 52-state proteins. That means, take the slow folding proteins, right, these proteins have more contacts than the fast folding proteins, because if you see the slow folding proteins the values are very high compared with the fast folding proteins, right. This we can see the relationship between the slow folders, fast folders and the contacts, right. They can have a picture related with all these parameters. Okay, these all we did with the completely with 3D structures. Now, is it possible to get this information from the sequence, because topological parameters, contact order, long range order, multiple contact index, we need a structure. It also depends upon the type of the protein, alpha, beta, mixed class proteins, right. Can we predict or can we relate the sequence based parameters with the folding rates? Last class we discussed about the stability, right. So, how many properties we used last time, which type of properties? Physical, chemical, energetic and conformance properties, right. We discussed about all the properties, they are available on the web, right, the original values are there. So, how can we relate these properties with the folding rate, right. There are various aspects to predict the folding rate from the sequence. Either we get the sequence based parameters, right. What are sequence based parameters we discussed in the earlier classes? Amino acid composition, occurrence, average property values, conservation, right, hydrophobicity profiles, right. So, we can see if we have many properties, you can see the average property values. If we take a full protein, right, you can get the average property values. How to get the average property values? Calculate total divided by like normalize with the chain length. So, then we see whether it can be related with the folding rate, or you can do, because contact order and long range order are based on contacts. If one program can predict the contacts, then we can use that contact to predict the folding rate, right, that is also possible. Or we have to model the structures and then we have to use the structures to get the contacts, because we long range order depends upon which atom? C alpha atom. So, crude model is enough, we do not need the refined model. So, then we can calculate the LRO and we can predict the folding rate, right, with the various ways. So, let us see whether any specific property, right, can relate the folding rate, right. So, in this case first we calculate the average value, right. For example, if we take the hydrophobicity, how to calculate the average hydrophobicity? Here we take the hydrophobicity of values, all the residues, right, j equal to 1 to n, right, j equal to 1 to n. Where is n? N is the number of residues in a protein, right. So okay, I can put n. So then normalize with n. Then we get the, for any protein I, we will get the average value. So we have the, for example, we have the 100 proteins. For all the 100 proteins, we can calculate the average property, right. You can give the average property here. So here P average. So here we have the ln kf. Now we relate. How to relate these two? Using correlation coefficient. You get the correlation coefficient, right. Then see whether any correlation between the average property value as well as the folding rate. So this is the data. So we used various properties, right, physical properties, chemical properties, conformation properties, energetic properties, and these are the properties which show the highest one. The values are very less, that is only 0.38. That means any single property they cannot relate the folding rate, right. That is also correct, that is fine, because if a single property can do it, right, then there is no use of these topological parameters and so on. Then we try to a secondary structure. What are different secondary structures? Alpha helices, beta strand, and the coil turns, right. You can see the contents, how many residues or the percentage of residues in alpha helix or the beta strand or the coil, right, or helices and strands. So we, if you compare all these secondary structure information, we can get up to 0.52, and then we use solvent accessibility, right. Either you can predict the solvent accessibility from sequence, or you can use this exact data, right. So what are different locations based on solvent accessibility? Exposed, buried, partially buried, and partially exposed, right. So we try to use various aspects, even the correlation is up to 0.4. That means amino acid properties or secondary structure or solvent accessibility, there are some limitations, so they can get up to 0.5. So on the other hand, if you use a contact order or the long range order, we can get up to 0.8 or multiple contact index, right. This, from this analysis, what can we infer? We can see that native state topology is very important, and that is a major determinant in the folding rates of these two state proteins. Okay. So native state topology means this will tell you the folding type of a protein, right. So in this, if so, what are the different types of classes? Alpha, beta, and mixed class. That mixed class means containing both alpha and beta, right. So if you classify the proteins based on structures and then use these properties, then we include the some of the topology aspects. So because for different classes or different folds. So then we did that. So in this case we see that all alpha proteins, there are some specific parameters, like the conformation parameters are important in all alpha proteins. The all beta proteins mainly thermodynamic properties, they influence the all beta proteins. The mixture proteins, we could see that some physical chemical properties, right, they play an important role in the case of mixture proteins. So if you classify the proteins into alpha, beta, and mixture class, then we could see that the correlation enhanced, right, very significantly, right, from the using all the data sets together. Now how to relate, right. In this case we have the folding rate values, and we use a multiple regression technique, right. We combine the different values, and then you can relate to the folding rates. Now the question is, when you make the combinations, there will be millions of combinations, right. For example, if you have the 5 residue combination, right, you have 2 residues means only 2. If you make more number of combinations, right, there will be lot of combinations. So then how do you see whether any particular combination shows the highest performance? So we did with the different combinations and see that this is the frequency of combinations. Most of the combinations you can see this is very the performance is less, right, and here this is the average performance more than 25%, about 25%. In the case of all beta, here also you can see only for a particular combination it showed the highest value, right. In this case we could see one specific combination, for example all beta, these are the properties, and for the case of the mixed class, these are the properties to get the highest correlation. Okay, from that you can derive equations, right, for all alpha, because less number of proteins, so it can be derived from just one property called alpha C, helical contact area. All beta proteins, they use 3-4 properties, and the mixed class 1, 2, 3, 4 properties. If you use these equations, right, then you can get the correlation of 0.9 for each of the classes, and the deviation is also comparatively less, and we checked with the t-test and the p-value, so the values are also statistically significant. Once it is done, then we can develop a program, right. So, this is a relationship between experimental and predicted values. So, you can see the good correlation, because you can see most of the proteins, right, the values are on the 45 degree slope. So, now if we get this, then we can make a server, right. So, here it accepts the amino acid sequence, single letter code, and then here the important thing is we need to give the structure class. If you do not use a structure class, if it is unknown, then it can predict the folding rate, but in this case the performance is not so good, and you can have some limitations if you use the unknown values. But if you know the structure class, whether it is all alpha or all beta or mixture class proteins, then you can take the particular class and appropriately use the equation, and this all alpha proteins, and you can see this is 8.44 per second. If you look into these structures, how many structures are known in the PDB? 1,30,000 structures are known, but if you take the folding rates, we know the folding rates only for about 100 proteins, 100 plus proteins. So, many proteins we know the structures. So, in this case we know that it is all alpha or all beta or the mixture class. Even with the known sequences, right, many sequences they have high sequence identity with the known structures. In this case you can get more number of sequences with known structure information. If the structure information is known, then you can use the server to predict this folding rate even if the structure is not known, right. So, you can use the sequence information. This is one section, you can use the amino acid properties combined together, and you can predict the folding rate. So, second aspect, for example, if you have a sequence, like how to predict the folding rate. So, there is an indirect method to predict the folding rate using contacts, because we know that topological parameters are important, right, and these parameters explain well about the folding rates, and the parameters are mainly depending on contacts, and if we are able to get the contacts, then we can convert these contacts into folding rates. So, these indirect methods they developed in 2005, right. Burkhard Rose from Columbia University proposed this one. So, what they did, so first they get the sequence, for the sequence they get the contacts. How to get the contacts? They use this amino acid sequence information. For example, so take any of this residue, for example take this valine. They have identified the residues which are close to valine, right. For example, this leucine, threonine, lysine, as well as on the right side towards N terminal and towards C terminal, and they located the position of this valine based on secondary structure, whether it is helix or strand or turn or coil, and based on the solvent accessibility, it is buried, this is secondary structure, and a solvent accessible surface area, right, buried or partially buried or exposed, and they compare with the known structures. Then they see this valine is in contact with which type of what are other residues. Take any cutoff, for example 8 angstrom, compare the experimental data, right. So, they use this information and they use the model for predicting these contacts. In this case they do not know the structure. For any sequence they give the sequence information, neighboring residues, and secondary structure, solvent accessibility, as well as the conservations, right. We have discussed all the the parameters in the previous classes. So, take all the information to predict the contacts. Once the contacts are known, can we predict the LRO? Yes, we can predict the LRO, right, because we see only we need the cutoff of 12 residues, then we calculate the number of contacts, residues with the cutoff of more than 12 residues. Once we get the LRO, then we can get the folding rate. Now, this is an indirect method to predict the folding rate using predicted contacts. Okay, here this is a server which can predict the contacts. So, here this is the amino acid sequence, right. You can take the amino acid sequence in single letter code. So, then they use all the other information. They use the what are information they use for the prediction? Secondary structure, solvent accessibility, and the neighboring residues, and the central residue information, conservation score, So, they take all the information, finally they give you the output. If you see here, if you take this is the 8 angstrom distance, this 8 means 8 angstrom, and these are the residues 67 and 99, right, 67 and 99 are in contact, right, with the probability of, this is the probability of 0.95. So, they give the probability, so you can take the contacts with a high probability, and if you get these contacts, we can convert this information to get this long range order. For example, if you see 69 to 99, what is the distance separation?
32.
33, right. So, you see I minus j 32. If you see this one, right, if you see this one I minus j equal to 8. Okay. So, we have all the contacts. Now, the next step is, once you download the data, we can get the I minus j and compute how many contacts are within the limit of 12 residues. It is very easy to do that. Now, the question is whether these contacts, right, are at least the residues with a separation of 12 residues, right, are related with the known long range order. If we have known the structures, we can get the long range order. Likewise, they can do that. So, they did that. For example, if they predicted the LRO, and this is the LRO obtained directly from the structure, and see the correlation. So, specifically if you see the small proteins, right, that they consider 199 proteins, and the correlation is not bad, is about 0.7. So, in this case, if you predict the contacts, and the predicted contacts help to get this long range order, but with the correlation of about 0.7. Now, they converted this information into predicting the folding rate, right. So, what they did, so they used all the predicted LRO and use the same equation and to predict the folding rate. So, here for a set of proteins you give the folding rate, right. So, here the correlation is 0.68 minus 0.68 with the deviation of 0.98. This outlier, right, this outlier mainly because it contains disulfide bonds, so it is not able to take into account. So, now if you exclude this outlier, then here the correlation is minus 0.78 with the deviation of 0.86. When you compare the data with the experimentally calculated the LRO, that means using known 3D structures, if the results there is comparable, is 78, this is 0.81. That means if you know the sequence and if you do not know the structure, indirectly you can calculate the long range order, and eventually you can get the folding rate from this long range order, right, which can be predicted from the contacts. So, this is how they can do that. So, if we summarize, right, what are the various aspects we discussed today?
Protein folding rate.
Protein folding rate, right. What is protein folding rate?
The rate.
It's a measure of how fast or slow a protein can fold from its amino acid sequence to its native 3-dimensional structure. So, what is the order of magnitude of these folding rates? Slow to fast, maybe in the microseconds to an hour. So, it also varies up to 8 orders of magnitude. So, if we compare with the all alpha proteins and all beta proteins, right, which are fast folders, right, all alpha proteins fold faster than all beta proteins, right. Right, if you can compare this information with the contacts, if you compare the all alpha proteins and the all beta proteins, which one have more number of long range contacts?
All beta.
All beta proteins can long range contacts, and also these contacts are influenced with which residues? Hydrophobic residues. The case of fast folders, you can see mainly the medium and short range interactions, and some of the polar residues are dominant. So, that is related with the folding rates, right. Then we discussed about different topological parameters based on the 3D structures and contacts, right. What are the different parameters we discussed? Contact order, long range order, and the multiple contact index. So, if you see these indices, all the indices, right, they showed good relation with the folding rates, right, about 0.7 to 0.8, right, inverse relationship between any of these indices like contact order or long range order or multiple contact index with the folding rates. This will tell you the topological parameters are important for determining the folding rates, right. Then we discussed whether the sequences or any properties can relate these folding rates, right. When you compare the sequence based information, secondary structure and accessibility, right, as well as the topological parameters, right, we found the difference among all these features. The topological parameters could explain up to 0.8, but in the case of the other parameters like the properties or secondary structure or accessibility, right, up to 0.5. Then we discussed about whether it is possible, whether we can predict the folding rate from sequence, right. Is it possible to predict the folding rate from sequence? There are various methods available, right. Either you can directly use the sequence, or you can indirectly use the sequence to predict the folding rate. How we directly predict the folding rate from sequence?
Physicochemical.
So we can use the physicochemical properties, right, because we derived various parameters, right, and I will discuss in the earlier classes. So you get the average values of these physicochemical properties, and we can combine together, and using linear regression we can predict the folding rates, right. Indirectly, how can we predict? Predict the contact first. Contact information we convert it to the indices, right, for the example the contact order or long range order, and use that information to predict the folding rate. So that is fine. So we have the standard equations, we can predict the folding rates. Fine. So we discussed the applications of sequence and structure based parameters on various aspects like structure prediction, protein stability and the folding rates, right. In the subsequent classes we will see how these parameters can also be used to identify the binding sites in complexes, for example protein protein or protein RNA or protein DNA or the protein ligand complexes, and whether we are able to understand the recognition mechanism of how these complexes form and how to maintain this binding affinity, and how these parameters are useful in structure based drug design, for example to identifying the lead compounds or potential inhibitors, right, for any specific targets addressing any specific diseases. Then we will discuss about some of the steps you have to follow to develop any of these applications, right, for your projects or anything, right, that we will discuss in the next few classes. Thank you for your kind attention.